Hey Lars, can you hear us?
Hello.
Hey, we can hear you properly.
Okay, great. Good to see you all.
Thank you.
Sorry for not being there.
Hi Lars.
Hi, that's all.
Yes, hopefully soon. It's still not 3, so.
The remainder. You're sharing the screen already?
Yes. Yes, I think they. So this is my laptop and this is the Zoom call, so I should be able to. Okay.
Béatrice est là, hein, c'est Angela qui.
C'est Angela.
Je pense que le jury reste valide. Oui. Il n'y a pas d'urgence sur les HDR, c'est ça. Est-ce qu'on donne une deadline? Je me connecte là pour vérifier.
Ah non, mais c'est s'il est capable de s'il a un channel pour te contacter.
Ah, t'as écrit le mail. Je peux me connecter sinon.
C'est le même problème. En
Ah oui,
fait,
non,
c'est
je
quand
verrai
c'est lui
pas
qui a
plus.
dit. Voilà,
Je
c'est
verrai
quand
pas
c'est
plus.
lui qui a dit. Il fait pas mal. Ah, c'est pas grave.
Donc, je ne vois pas.
Ah, incroyable.
Voilà. Universel, c'est ça.
Hello, Angela. Can you hear us?
Yes, I can.
Okay, perfect.
Apologies for being late. I think it was a time zone error.
No problem. Thank you for joining.
So, welcome to this defense. I'm Pierre Twangenbaum, one of the members of the jury. I'm a research director at CNRS, Natural Language Processing. And if you all agree, members of the jury, I will be chairing this defense. No opposition. I'm a local, actually. That's why I'm candidate for this. We will have first a presentation by Raphael for 3/4 of an hour, approximately. Then the members of the jury will ask questions, and I'll give you the floor, starting from the most remote rapporteur first, and then other members of the jury. Then we will have a private session where we discuss the results and prepare a short report. But in the meantime, Raphael, you have the floor.
Okay, so thank you everyone for joining me today, especially members of the jury here and remotely. So today I'm gonna present my PhD thesis, which is called Joint Representation for Heterogeneous Image Sets and Text. And I've been supervised by Bertrand and Damien. Okay, so a quick outline of the presentation. So we're going to start with a cognitive mapping of the human brain starters, and then we'll dive into meta-analysis of neuroimaging data. And then we'll go through three contributions of this thesis. The first one, which is called MNODE, which is large-scale decoding of statistical fMRI data. The second one, which is called Pix-to-Image, which aims to give dense spatial representations for automated meta-analysis. And the third one, which is called Neurocontext, which explores a contrastive text-to-brain mapping for neuroimaging data. So first, let's look a bit into mapping of the brain, and we focus particularly in this thesis on task fMRI data. And the idea of task fMRI data is that we're going to ask some subjects and patients to perform a task or undergo a specific stimuli. And we're going to record the BOLD signal, so the blood oxygen level dependent signal, which is a proxy for neuronal activity. So we expect in regions which are related to the task that the signal will have a shape, which is called the hemodynamic response function, which is shown in the figure. And what we're looking at is, okay, is this response active in areas? But we want to specifically know the areas which are specific to this task. So we are comparing this response to the response we obtain from a control condition, which often eliminates some of the elements that we don't want to look into. So in this example, so the task we look into is listening to a sentence, and the control condition would be reading a sentence, and we would avoid, for example, looking into how to process information from that given sentence. Then, so from these BOLD signals, there's a lot of preprocessing steps which are not detailed in this thesis. We rely heavily on already preprocessed statistical images that are obtained from those experiments. And how do we report the results? They are usually reported in a shared space, which is called the MNI space. There are other options, obviously, but the MNI space has become kind of a center for and a reference for reporting those specific areas of activation which are related to our task. We're going to also talk a lot about decoding and encoding, which are the two processes of okay, from a cognitive concept, can I know which areas of the brain are involved for this process? And so this is the encoding, and the reverse task, which is decoding, which is okay, if I give you a specific activation of the brain, can you tell me which cognitive concepts, which cognitive aspects are related to it? And so there are lots of experiments which are shared in the world, but they have limitations, and mainly due to the acquisition cost. So it takes a lot of time plus the MRI scanner use. So they are often restricted to a few cognitive processes. So neuroscientists will tend to focus on specific processes. And additionally, the number of subjects are often limited, which induce a low statistical power in the results that are shared. And we'll talk a lot in this thesis about meta-analysis, which is a way to aggregate results that come from different studies. And what we look into specifically is to find consistent effects for a given process across this literature. And what we want is to put those processes in perspective, to build a cartography of the brain, a cartography of the cognitive processes across the brain. So if we look a bit into meta-analysis, so as I said, the goal is to aggregate studies and build a consensus across publications that are shared, results from neuroscientists that are shared. And those results, they come typically in two modalities. The first one, which are the unthresholded statistical images, which lead to image-based meta-analysis. So we are going to aggregate from those statistical images directly. But the main source today is and are stereotactic coordinates, which are contained in publications. And we see here the process of extracting those stereotactic coordinates. So instead of publishing the full statistical image, which is quite heavy, and especially maybe not today, but in the past it has been. So the process is usually to threshold the statistical image that we obtain from the results, from the Bolt signal experiments. Then we're going to identify some cluster of activations, and we're going to extract the maxima that we find in those clusters. And typically we find in publications what we call coordinate tables, where we have in this MNI space, in this shared space, we were going to have the coordinates of those maxima. So obviously it's been shown that it's better to use statistical image to perform meta-analysis, but the current state of the community is that a lot of the activity is shared as peak locations. So there is a lot, we will talk a lot about coordinate-based meta-analysis in the rest of this presentation. So the process of meta-analysis is to aggregate those reported activations. And so there are usually 3 steps to perform this. First, to collect some studies, which report, for example, here, we're going to take publications that are related to the reading term, which each of them reports some activation in those coordinate tables. We're going to aggregate those locations on the same level, and then we are going to perform some smoothing and some thresholding, which is going to give us the consistent effect that we have across those many publications that look into the term. Usually there are 2 kernels that have been used heavily. The first one is a Gaussian kernel that we put around the location of each reported activity, and which reflects the uncertainty in the reported peak position. So earlier I talked and I didn't talk about all the heavy preprocessing steps that are involved to generate the statistical image that we study. And all this preprocessing that we apply to the Bolt signal induce some variability, which has been shown, like from team to team, the results that are reported can be a bit different. So we induce this uncertainty either through a Gaussian kernel or either through a kind of constant kernel around the peak locations. So to perform this meta-analysis, we need to have access to publications quite easily. And in the communities, there have been a lot of efforts to make this access to the publications quite easy. So it started all with a brain map, and then Neurosynth collected some publications, and now we have some tools like PubGet, which aim to parse the PubMed API quite easily. And what we observe is that there is a constant number of publications that are still published each year. In particular, we focus here on the publications that contain stereotactic coordinates, and we see that there is 1,000 a year that are published. So if we want to look into automated meta-analysis, and because what we aim here is to build a cartography of the brain, there are several issues to deal automatically with such a large number of publications. The first one is, okay, can we extract the results from the studies efficiently? And there are tools like PubGet that does it perfectly. And the other one is to capture the nuance of the experiments that are contained in the publications, so properly handle the text that is available to us. So if we look a bit into automated meta-analysis, the first and the reference remains the neurosynth model, which has collected a dataset of 14,000 publications, making all the abstract and the activations that are in the publications easily accessible. And we can see it as an automation over the brain map project. And what neurosynth does is that they perform a chi-square test over the term occurrence in the publications. Okay, so we're going to look at publications that report language versus publications that do not cite the term language. And from this, we obtain this image, which is the result of the chi-square test of independence. Some limitations remain. So the independence test is performed on each voxel separately, which is a bit, so we know that brain activity is correlated in some way. So performing the test independently might be limited. And also the in-sample testing does not assess for generalization. So if we look into the neuroquery model, there is a shift from neurosynth to neuroquery, which is predictive meta-analysis. So instead of looking exactly at occurrences inside the publications, neuroquery is able to handle a wider range of queries by typically projecting the queries that the user passes onto a set of known terms. And how they do this is by leveraging the term frequency inverse document frequency matrix, which makes rare terms become more significant. So in the independence testing that we have in neurosynth is unable to perform such queries on rare terms. So here, by mapping any term from the vocabulary to this set of known terms, we are able to predict a statistical activation by combining linearly those reference maps. So one limitation of the TF-IDF is that it fails to capture the semantics of the query. And so neuroquery is not really adapted to handle large and complex queries. So recently we've observed the rise of language models, and text-to-brain base themselves onto those language models by leveraging a SciBERT, which is a BERT model trained on scientific data, to compress the textual information from the text into an embedding space, which is appropriate to, on the other hand, predict brain activation from this compressed representation of the SciBERT model. And this is done by passing the SciBERT embeddings through a deconvolutional network, which is able to reconstruct brain activation from it. So there are some changes that's present in the automated meta-analysis field. And the first one is that there hasn't been some major focus on image-based meta-analysis recently. And the question is, first, do current image databases are able to support this image-based meta-analysis? Then the question that rises is, okay, we have this amount of images that are available. Can we combine a coordinate-based method with image-based method to create a middle between CBMA and IBMA? And the last one is, okay, text-to-brain raised the SciBERT model to handle the text of the publications. Can we improve of that and leverage more recent models and LLMs to improve the meta-analysis process? So we tackle those in the following contribution chapters. So first, the unknown model leverages NeuroVault, so a large-scale image database to perform multi-label decoding of statistical images. Second contribution is Speech-to-Image, which tries to combine CBMA and IBMA all together. And the third one leverages large language models in a contrastive setting to better model this meta-analysis process. So decoding, which is associating cognitive concepts directly from a statistical image, It has proven effective, but it's often limited because usual decoding settings in the literature are a multi-class setting. So each statistical image is only associated to only one cognitive concept, which is a kind of a reduced view of brain activity. And the second one is that it's usually a within study setting. So there is not a large generalization that is able to handle new samples. So we address in the model those limitations. First, by using a multi-label settings. So instead of associating only one concept to a statistical image, each image will carry different concepts. And also, we show that we are able to generalize to new collections, to newly acquired data that are unseen before. So how do we do this? First, so we train a decoder in a multi-label setting. So the first step is to collect images. And what we do is that we collect images from NeuroVault. And what we are able to collect is around 50,000 images, which are Z or T maps, that we standardized and that we compress through the Diffumo Atlas. Okay, so instead of having the full statistical voxels image, which carries thousands of voxels, we decompose the image onto a dictionary, which gives us a vector of loadings, which is usually 1,000 components, and which considerably reduce the dimensionality of the problem. On the annotation side, what we want is to have for each statistical image a set of cognitive concepts which are already relevant to this image. So what we leverage is annotations from NeuroVault, but the issue is that annotations in NeuroVault are left to users, so they are quite open and there are no standardization from a collection to another. So a collection being results and images studied by a given neuroscientist. So we handle this by performing a mapping of some labels through regular expressions, and also through an ontology-based association, such that parent concepts will apply to the images. And we publish, obviously, those data on GitHub so that it's freely available. What we show first is that we successfully decode around 60 concepts, which is way larger than what the current number of classes that we've seen in the literature. And we successfully decode most of them better than other methods from the literature. But still, there are some concepts that fail, such as working memory, for example, which cover a large number of underlying tasks. And that's why for those specific contests, we fail to have a specificity in the statistical images that help us identify them properly. What we observe also is that, we play a bit with the Diffumo Atlas that we use, because it comes with different resolution, ranging from 64 components to 1024. And what we observe is that the more precise the decomposition is, so leveraging a larger dictionary size, the better results that we obtain. And especially what we do is we tried stacking the different resolution to obtain a different precision in the patterns that we observe. And we observe that stacking outperformed other individual representations. We also eliminated negative components. So the Diffumo Atlas is a positive, strictly dictionary. So by eliminating the negative components, we kind of erase areas which have negative statistical values, and which might be linked to the control condition. And by doing this, we obtain a specificity on elements that come from the target condition and help us identify better the underlying process. What we observe also is we look into the sensitivity maps that we obtain after we train the decoder. And what we observe is that the sensitivity maps that we obtain for well-decoded terms are coherent with what we could expect by looking into the literature. And when we look at concepts that fail to be decoded properly, we observe no specific pattern, which lead us to think that the learning didn't went great. And this is interesting because this is kind of where we see the link with image-based meta-analysis, where we can properly identify patterns which are related to cognitive concepts. By looking into those sensitivity maps, we also identified some bias in the data. So in particular, what we looked at is emotion perception tasks, which reveal the face, the FFA, which is responsible for identifying faces, which make us think that, most tasks that are involved for emotion perception are actually people looking at faces having some emotions on it, and which can be a problem. And it shows also an underlying problem is that data that we collect from NeuroVault are coming from different collections, so different work from different labs, and some labs have published tremendous and large collections such as HCP, and they kind of we see the bias of experiments coming from those large collections onto the results. So what we show with Zenode is that we can use large-scale databases for meta-analysis, and we can do so by showing that we can decode the decoding performed well over a large set of cognitive concepts in a multi-label setting, and it also generalized to unseen data and unseen subjects. What's interesting is that we particularly looked into the sensitivity maps, which show coherent pattern with the literature and with what we could expect on specific cognitive processes that are well decoded. But obviously, the annotations that are available in NeuroVault, they are a limitation by their nature because it's ranging in different formatting, and there is no unification across collections. So what we looked after this is on the other side, in coordinate-based meta-analysis, we have peak locations that are reported as stereotactic coordinates, which are kind of poor representation of the activation, of the activity that we have in a statistical image. So could we, in some sense, use those large database of images to inject some knowledge into the coordinate-based meta-analysis process? And what we do is that we leverage images to perform the reconstructions and to form a set of peak locations, try to reconstruct the statistical image that induce this set of peaks. So to do so, we use the set transformer architecture, and for two reasons. The first one is that in each publication, we observe potentially a variable number of peaks that are reported, and this architecture is able to handle this variable number of elements as input. The second one is that still in those publications, the peaks that are reported, they come with or without statistical significance and statistical value. So there is no specific ordering we can induce on the peak that are reported, and this set transformer architecture is permutation invariant, which makes sense in our case. So in practice, what we estimate is are the DFUMO loadings, so the 1024 vectors that represent the image. And we can obtain, obviously, the image by linear regression over those DFUMO loadings. And those DFUMO loadings are coming from the set transformer, which under the hood perform an embedding over each peak locations and then aggregate all those peak representations. We are in a supervised setting. As I said, what we leverage is neural vault images from which we extracted peaks, and then we obtain a set of matching peak and image so that we can train this model in a supervised manner. The way. So first, to evaluate this reconstructed model, we leverage the dice measure instead of correlations. And the dice coefficient, what it measures is the coactivation of voxels upon a specific threshold. Okay, so what we do to compute the dice measure is that we have two given statistical image. We're going to threshold them to the top 5% values or the top 10% values, and then we are going to just denote as being activated or not. And then we are going to look at the intersection over the union to evaluate and to compute the dice score. So ideally, the dice score of 1 would mean that there is an exact match between the two images. And on the opposite, 0 means that there is no intersection over the activated voxels, which wouldn't be ideal in our case. In the other way. So we show that the pixel image, we left out neural vault images, which are kind of ground truths, from which we extracted the peaks. We are able to obtain better reconstruction metrics here compared in particular to the kernel density estimation, which is placing just a Gaussian kernel around each peak location. We also compare to Pix2Maps, which is a UNet architecture trained also on neural vault. And what we show is that we outperform both of them with quite a margin. So once that we have this reconstruction model that is trained, what we can do is reconstruct the literature, the neuroscientific literature. So what we do is that for each neuroscientific publication, we extract the activation table that we have in it, and we pass it through this set transformer to generate a DFUMO representation of each publication. By doing so, we can perform similarly to NeuroSynth. We can just perform a statistical testing based on term occurrence and build an atlas from those reconstructed literature, and we obtain the Pixel image atlas, which is published. And what we see is that we obtain patterns which are relevant in our case. But what we do additionally is that, okay, can we use now this enriched literature to perform decoding on real images? Because that would be an ideal case, because we have a large amount of textual information in the literature, and if we can use it to address real-world statistical images, we would be able to considerably enlarge the vision we have of cognitive concepts that we can attach to each statistical image. So what we do is that we extract labels from each publication text, and we train a similar architecture of decoder based on only the literature, and we adapt and we apply this decoder to NeuroVault without supervision. Okay, so the labels that we have are automatically retrieved from the literature, and there is no painful annotations that we have to go through for each statistical image. And what we show is that, okay, this reconstructed literature is a solid base for image decoding and on a wide spectrum, okay? So what we show here is that we aggregated each task from NeuroVault by cognitive atlas category. And what we show is that across all categories, we obtain results that are above chance by a margin, showing that, okay, indeed, by leveraging the literature, we can perform decoding on images, and there is no supervision in the training data that is necessary for this. An additional point is that, okay, we evaluate on NeuroVault and on those particular tasks because those are labels that are attached to NeuroVault, but the vocabulary that we use is 6,000 elements. So we could potentially predict other cognitive concepts, and we are limited here by the NeuroVault evaluation. We also show that we outperform other representations. So if we use the kernel density estimation or the Pix2Map version of the literature, and we perform the same decoder training, we obtain lower results. And also, we obtain lower results if we look at coordinate-based methods such as NeuroSynth and NeuroQuest. Once we did this, we attempted to do some variants of the initial Pix2 image model. And the first one being, okay, so in this model, we still use task fMRI data to train the reconstruction process. And the question is, how could we alleviate this need for task fMRI data? So what we did is we performed ICA on resting state data, and we use the components that come from those ICA runs as training data to our model. Another attempt that we did is that instead of using the Diffumo representation of each image, we use an autoencoder which is trained on NeuroVault to see if predicting a latent space of the autoencoder predict more useful patterns than predicting the Diffumo representation of the task fMRI data itself directly. So what we observe is that, okay, we have similar reconstruction level, in particular for the autoencoder case, which has been trained itself on task fMRI data. So it's just a different dimensionality reduction technique. And for the ICA version, so in purple, we see that the results are a bit below, so they still quite outperform other representations such as the kernel density estimation. But it's still a bit below the task version. In particular, if we take some time to analyze the ICA version, the reconstruction results are unequal in the brain, and in particular, there are some regions which specifically suffer from this reconstruction process, which are visual attention areas and attention areas. And we observe that, okay, this has influence over the decoding performance of the decoder that is, okay, I'm allowed to update. Okay. And what we see is that on attention categories related tasks, okay, the decoding performance that we obtain are way worse for the ICA version than the task one. Okay, it's painful. Just gonna quickly, sorry about that. In practice, so what we observe is that the ICA version tends to produce more blobby activation, and it fails to capture co-activation patterns. So there are some regions that should be activated that are not activated. So what we show with this image is that we can bring coordinate-based and image-based meta-analysis all together. Okay? We provide dense representations to coordinates, and what we show is that using those dense representations, we can build an atlas which is based on reconstructed data. And second, that we can perform decoding on images on a wide set of terms and without any supervision. An obvious limitation in those days is a TF-IDF-based labeling, which has been quite outperformed by LLMs recently, and we address it in the following contribution. So if we look at the way that models extract information from publications right now, we have NeuroSynth, which look at the exact word occurrence in the publications. We have NeuroQuery, which leverages TF-IDF, which gives us more importance on specific terms, on rare terms. And we have TeXtBrain, which leverages the Cybert model, but only on the abstract due to the length of the input, the content length of the model, which is not enough to handle the full publication of the full text of the publication. So the question is, can we provide better representations for those typically large inputs, such as scientific publications? So we built the neurocontext models, and one key component is that it breaks with the usual, okay, let's perform decoding in one way, encoding in the other, by generating a latent space, which we can use for both. So what we do is that we use the Mistral model to obtain fixed representation of the literature, and we, instead of restricting ourselves to the abstract, we chunk each publications, and we aggregate those chunks by a mean over the embeddings. And then what we do is that we align the representation we get from the text with the representation we build from the reported activations that we have in each publication through contrastive learning methods. What we do is that we evaluate on left-out publications this ability to match the proper text with the proper activation. So we use a recall, which is on a pool of 1,000 publications. If I give you a text, are you able to retrieve in the top 10 the given activations that goes with it? And what we show is that, okay, so first, we outperform other methods such as NeuroQuery and TeXtBrain. But what's also interesting is that methods that such as NeuroQuery and TeXtBrain are not able to benefit fully from using the body of the publications, which is the full text, the full content of the publications, where we are able to still gain from the content that is beyond the abstract. We show that, okay, first, one brick is the contrastive learning part. So we tried, instead of using contrastive methods, directly to directly predict the proper activation. And what we observe is that adding a contrastive latent space to the model clearly benefits to the results. We also looked into different LLMs to evaluate the impact of them, and in particular the impact of the number of parameters that they contain. And we see two elements. So, for example, there is a first difference between Cybert and GPT-Neo 1025 million. We see that there is no particular gain, and the gains come afterwards by adding parameters. But what's underlying is that the embedding space is larger for those models. Okay, so it seems that the larger embedding space that we obtain from the text helps to better capture the nuances that we have in the publication. So on this latent space, what we do next is to train an encoder. So we want to predict the brain activity from this latent space. And what we do as an evaluation task is that we took the description that we have in IBC, which is a collection of task fMRI data. So we took a description of a task in IBC, and we encoded those elements into first the text encoder, and we pass them from this latent space, we pass them to generate brain activity from it. What we observed was quite deceiving in the sense that we did not outperform other methods. And one hypothesis that we have is that the IBC contrast definition is typically 100 character long, which typically falls in the regime where other methods perform well. So we think that contrastive learning is a promising avenue to handle the full content of the publications. First, because it shows that we can benefit largely from the full content of the publications instead of restricting ourselves to the abstract or a part of the text. But our models still fail to properly exploit short text. So that's why we are investigating current issues with this model, along with Fatima Gayem from the team. And so those first 3 points are kind of our current work. First, so the encoder is trained a posteriori from the latent space. So our guess is that it makes it harder for him to benefit from the learned latent space. Second, there is a shift that occurs between the publications that we pass during training and the embeddings that we get for short prompts, such as typically IBC contrast definition, and which probably hardens the downstream task and the downstream evaluations that we did on ourselves. And also one last point is that currently we perform an aggregation which is the mean over the chunk that come from a single publication. And we experimented by learning, for example, aggregation through a transformer-based method, for example. And this mean outperforms learned aggregation. So we are curious to understand why exactly. So in this thesis, so first, we showed that we can perform IBMA with current image databases, and we can do this with minimal curation and in a multi-label setting. And this is important to really capture the variety of processes. Then we showed that, okay, those image databases, we can bring them all together with coordinate-based meta-analysis by providing a dense spatial representation to those publications. And it enabled us to perform decoding on a wide set of terms without any supervision of the labels. And then we showed that we can better summarize the literature with LLM and through contrastive learning. And I guess this is a promising avenue because what we want in the end, now that we have made the link on the coordinate and the activation side, I think the main avenue is to better get information that are contained in the literature. So from this thesis, there are 3 publications. 2 of them, one of them you published in Nature Scientific Reports. One of them was accepted to MICAI yesterday night, and the other one is under review. And all the code is published online on GitHub. And I would like obviously to thank my supervisors and also collaborators. So Fatima is in the room, and Jerome who couldn't make it this afternoon. I'd also like to thank Ross, who welcomed me for several weeks in Stanford in the team, and obviously to all the members of this team, which makes it really enjoyable to do a PhD here.
Thank you very much. Thank you very much for this very clear presentation, but in time also. So now I'll give the floor to our jury members, starting with Professor Angela Laird. So Angela is a professor at Florida International University. Angela, you have the floor.
Hi there, can you hear me?
Yes.
Yes. Good.
Wonderful. So first of all, I want to congratulate you. This is a remarkable amount of work. I am truly impressed with the quality of this dissertation. The computational and semantic challenges, I've spent lots of years with this data. I know that it is very difficult to work with, and the types of associations that you're drawing, it's hard work. So I'm very appreciative of all of the hard work that went into this. Your presentation and your document was very clear and well structured. Sometimes it can be hard to take such big topics and compress them down into a digestible format. So again, well done on that. I want to start with a question about the NeuroVault analyses. I'm particularly interested in NeuroVault. I do think that it offers a whole new modality for meta-analyses in neuroimaging. So much of the field has relied on coordinate-based meta-analysis, and not enough people have taken advantage of image-based meta-analysis using NeuroVault. And a lot of that, I think, is due to the nature of the data that's in NeuroVault. Our experience has been that NeuroVault data are very noisy. I have a graduate student who has spent the past several months, I want to say even 6 months, trying to curate these data. There's a lot of incorrect annotations by the users. There's wrong modalities. So you think you're looking at bold maps, but you're actually looking at connectivity maps. There's duplicates. There's mistakes in contrast. So I was just curious if you had similar observations. How did you address this? What was your curation process? Did you spend a lot of time curating NeuroVault? Or did you try to just take what was there and from that noise draw some signal out of it?
So the NeuroVault curation is a topic indeed. So we implemented a lot of kind of rules to remove anomalies that we find in NeuroVault. So typically maps where it doesn't coverage the full brain, or maps that have abnormal values in it. As you said, we also noticed that there are some duplicates, and I think some of them are from us because there are multiple collections of IVC, for example. So we kind of, at least on the image side, it's quite easy to maybe not easy, but we can visualize what's going on, and it's easier to implement some rules to rule out some images. What we end up with is 50,000 images, but there are way more images that are available from on NeuroVault, and I think it ranges around 200,000 images. So we implemented quite some pre-processing, and we kind of published this pre-processing online. So it might be worth to look into it. The pre-processing pipeline is published under the MIND-INRIA GitHub repository. But yeah, it's definitely a lot of steps, a lot of computational power to do this pre-processing. So I don't have a. I agree with you on this point that it's a tricky operation. On the annotation side, it's kind of harder to automate decisions. So what we did in particular is this cognitive atlas-based assignments, where what we kind of enriched the annotations that are associated to each images by giving parent processes and parent element from the cognitive atlas ontology to those images. So it doesn't solve the fact that some annotations which are associated with images are wrong, and
right.
Unless trying them one by one, you are kind of stuck but at least for some elements, it helps to build a larger view, a larger context of annotations which are associated to each image.
Okay, so let me ask you this. You worked with a lot of different data types. You worked with a lot of different databases. NeuroVault has data, but it's messy and noisy. But images are better than coordinates. You moved things along with respect to the coordinates, but take a step back from what you've done so far. If you were a user, so you've done a lot of methods work, but if you were a user and you wanted to do image-based meta-analysis, based on what you've seen in your data, your analyses, your results, what are you going to recommend to people who come to you and say, hey, I want to do an image-based meta-analysis. What's the best plan for this?
Say good luck. But more seriously, so it's something actually we discussed when I was at Stanford. NeuroVault has this downside of everything is left to the user, but at least we have the data, which is kind of the necessary beginning to start somewhere. Because if you enforce some constraints, some rules on what you need to do to upload stuff, people are just gonna not upload stuff and stick to the coordinates, which is kind of we can do better now. What we did in our experiments is that we focused on particular collections which are known to us, at least, to be more precise and more rigorous. So in particular, to train the pixel image model, we focused only on IBC maps, which may induce some bias in some way, but at least we know that so the training part is kind of critical to generating a good representation. So we try to really focus on images that come from IBC and which we know to be we can trust, and if we see problems, we can directly look and talk to members of the team which are responsible for the acquisition. But it helped us to get better reconstructions and to get a more let's say relevant model in the end. But if you need to perform a meta-analysis, I'm not sure exactly how I would do the collection of which images, which collections I'm taking into this meta-analysis process. Because as I said, so there is this problem of some of them being non-trustworthy. But there is also the fact that some collections are way larger than others. So it might be tricky to find a set of collections which have the good size and which are kind of equivalent in size, and so that the aggregation goes smoothly.
Do you think there's a way that the NeuroVault upload and curation process could be improved?
I think that something that could be done is that in the end, what you want to have a curated set of images is that you want to enforce some constraints on the upload. And what could be done maybe is a two-step process where you can either upload your data as is without filling the constraint properly, and one step further, which could be filtered quite easily in your vote, where you fill those constraints and you get a little badge, and you're happy with it. And but at least you contribute, and people know they can trust your data, and they can trust this subset of data in your vote, which could be a nice way to not eliminate the contributions to NeuroVault by not enforcing those constraints, but for people who want to build this this database, this proper database, and this rich database properly, to have a kind of a premium, premium set of images that you can access. I say premium, but I wouldn't ask for a fee to access to it, but just kind of more precise annotations attached to it.
All right, thank you very much. I will let the next person ask their questions.
Thank you.
Thank you very much. I'll now give the floor to Professor Lars Muckli from Glasgow, where he's a professor of visual and cognitive neurosciences.
Yes, hello. Can you hear me?
Yes.
Very well.
Okay. Yeah, very, very nice presentation. Very nice thesis. I was really enjoying reading it and learning. I'm using every year for my class the tools provided to do neuroquery and neurosync, and I know from experience, like they design experiments from areas in which I have no expertise, and then we use these tools to find what we would be expecting to find as a result, and it just using the peak activities and so on is quite impressive how it works. So somehow it works, at this level. So it's incredible tools that are provided. We spend a lot of time designing experiments very precisely with like parametric designs, MVPA analysis, and I wonder. This is now a question about, again, NeuroVault, which I don't have experience in using, but maybe you can help me a little bit, explaining how do these different ways of making maps get integrated. So let's make an easy example of, like, you have faces. I think you used the example as well, faces and emotional faces, right? And if you compare that to a baseline or to one another, that makes total different maps. So how's this used or reflected in the way you use the data?
So on NeuroVault, you have access to the annotations that come to with the image. So it's up to the neuroscientist to fill in for each image what is the experiment that led to it. So there is, I think there are multiple fields for that. There is a kind of a short tag that you can assign to it, and there are longer descriptions. And typically, so if you look at IBC, you will have short tags, and you will have those longer description, which
okay,
use a precise description of what happened, and I think that's a way we can at least separate some types, such as emotion versus faces. So again, go ahead.
Yeah, there's a conceptual match, right? So somehow you're capitalizing on trusting the interpretation of the data that is meaningful, and find the latent variable space that then reflects to those concepts that you're mapping.
If I may add on this, I think the best way, we actually looked into it, the best way would be, you have the publication, it has a DOI, and you fill the DOI in NeuroVault, and then you can, people can either look at both the publication and the images that are available, and in this way you you can have access to the exact protocol you have, and that's why extracting proper information from the text becomes crucial. So I think in the middle of my thesis, I looked into the number of overlapping images that have a DOI and which relate to a specific publications. And I think it was less than 1,000. I would say 200 or 300. Let's see. So I think if it becomes more automatic to fill this link, clearly there is an avenue to bridge the text and the activation altogether.
Okay, so I guess for the highest abstraction level alignment, it makes sense when you're comparing the peaks to the interpretation of a whole paper that they are somehow aligned on a very high conceptual level in a most useful way, let's say. But if you go one level down and you're comparing in NeuroVault these whole brain maps, how, for example, I mean, if you use a different technique like multivoxel pattern classifier, you could be looking at no change in activity, but being able to classify the content in a certain region. Would the map of classification be uploaded to NeuroVault, or the underlying basic activity maps for the different conditions? Just a information question. I mean, I'm not sure. I haven't worked with NeuroVault, so
I'm not sure I got your question.
Okay.
Be able to properly answer. I feel that I'm going to answer on the wrong way. So maybe could you phrase?
Yeah, I mean, so if you have condition A, B, and C, and they induce different amplitudes across the cortex, you could compare A to B, A to C, and that gives a good landscape for your interpretation. But if you're not comparing the activity maps, let's say, there's an area, the default mode network is always active, but in those areas, you then use multivoxel pattern classifier. So you use a different way of looking in, multivariate analysis to then find there are regions that are coding differently for A and B, but the overall activity is the same. Yeah, maybe.
Yeah, I think on your work you can upload different type of modalities. So it's not restricted to uploading statistical images,
Right.
And there are ways to indicate, okay, this is a T map, this is a beta map,
Right.
And so typically, I think this falls into your question where, okay, what you're uploading is not a Z map, and but you have to just fill in to indicate what exactly was the procedure to obtain the result.
Okay, thank you. That clarifies this. Very helpful. Now I have a question to one of the main figures. So in your thesis, I think it's A3. So you show the accuracy of classification on the different validation folds, but you have the great performance for emotion perception, emotion, mental imagery, motor planning, that come out very reliable, very at the top, and very, very poor performance for spatial ability, visual recognition, and visual object recognition. And I find these extremes quite surprising, because I would imagine that, for example, visual object recognition is a very standard, controlled way of doing experiments where different labs probably do the same way of analysis, whereas emotion perception and mental imagery can be very different. What's your explanation about this unfolding?
So I couldn't explain on the visual object recognition side. For example, for emotion perception, what we looked, I think the way to look at it is through the sensitivity maps. First, identify if there is a kind of bias or somewhere that is occurring. And for the emotion perception side, we saw that, okay, it is face area that comes into play, and okay, that maybe drives the cognitive concepts to be identified quite easily. What the trend that we observed a bit in the results are still that okay, so we do this parenting assignments. What we observe is that generally the larger categories and the higher in the hierarchy, they cover a wider variety of topics that are the children as they support. And that's why they fail to identify proper patterns with it, because okay, it's too large of a category, and it's not meaningful anymore and not specific anymore to be recognized. But for specific terms, for the visual object part, I couldn't. I would need to look into it, to look into the sensitivity maps and look into, in particular, are there some larger collections that induces an effect? Because we had issues, for example, so HCP is more than 20,000 images, so it's a very large part of our neural data set. And depending on how HCP is behaving, it can drive the results some way or the other.
Okay.
So yeah, so I don't have a strong answer to this.
Thanks. That's very helpful. Related, so in the main figure there's like working memory is the second last, and from I think this is already the enriched model. So my question here is, did you I mean, enriching model was introducing a kind of a hierarchy, if I understand correctly.
So the enriched model contains a hierarchy, and I think that's something we discussed with Bertrand 1 or 2 weeks ago. There is a kind of swap in the label around the working memory in HCP, which might induce the fact that we are indeed decoding poorly the working memory, but it's kind of a bug we need to fix in the data. And so yeah, it's for this specific categories, for this specific topic. Yeah, the issue might be a labeling issue rather than another element. Okay,
I was that's interesting. I was wondering whether kind of enriching unfolding the hierarchy is saying, instead of working memory, have visual working memory, auditory visual working memory, auditory working memory, or something like this would increase the chance of finding reliable patterns.
Yeah, I would expect so. Having more specification in the topic would induce better decoding performance.
And I was wondering, so this is a bit going a step further. So having seen this amazing work, I wonder where you want to go with it next, or what else you could be doing. And you know, maybe you want to say a few things about this. But one question I was thinking is, do you think you could use this neuron context mode in a way to search for new hypotheses, let's say areas that aren't very well described, or generate maps that are very reliable, but maybe not well described, or where areas in which the language description is very confused, or some kind of meta-analysis that kind of informs the field of what new directions to investigate.
So I think that's so the main focus right now is to do the link between coordinate-based meta-analysis and image-based meta-analysis, mainly because we want to have a rich textual information and a rich spatial information, and so that's why we focus particularly on bridging those to be able to. That's what you said is working memory is quite a small description of the reality, and the variation in the sentences and explanation around those working memory experiments are kind of crucial to understand what exactly is the neuroscientist looking into. So my opinion is that leveraging the text from the publication is crucial here to answer to this question, to have a clear view and a larger, broader view of the underlying images, rather than having small tags and small descriptive like this. And what you said about looking into areas that are not well studied and such, I think it's hard because it's still a meta-analysis process. So we are giving results that come from the literature. So if the area is not well processed, is not well studied, it's going to be hard to take some answers that do not exist. But typically I guess that for publications that are very restricted in numbers but still tackle a precise element, it might help to give those publications a larger voice, a deeper voice, I would say.
Okay, thanks a lot, and congratulations again. It was very stimulating to read your thesis.
Thank you.
Thank you very much, Lars. Now moving to Professor Beatrice Moerkerke from University of Ghent. Beatrice, you have the floor.
Thank you. I also first want to congratulate you with the work you have presented in your thesis. I think your contributions to the field of neuroimaging are substantial and important. And I also think you have done an impressive amount of work. And moreover, throughout your work, you have proactively anticipated and addressed potential limitations or extensions, which further underscores the robustness of your research. I found your thesis very well written, comprehensive, and a pleasure to read. And I think we also saw that today in your presentation, that your talent for providing clear and insightful graphical overviews of methods and concepts greatly enhances understanding and engagement with the material. So my first question maybe is a small question that links a bit to the discussion that unfolded before. And when you, in chapter 6, where you used the NeuroVolt database, you have encountered that in the decoding process, emotion perception was confused with face perception, as emotion perception is mostly tested by showing faces in the currently available data sets. And I think findings like these are important and can help shape a repository in a more balanced way. Do you think there are ways of doing that? Of course, realizing this is a first step, but are there ways to proactively further complement the repository in ways that keeps these issues in mind?
I guess there are kind of a pool of protocols that one can use, typically for emotion. And one could imagine that okay, you could indicate which protocol you used, but I'm not sure exactly how I would do so. I guess the best option is to through language models to get this information. Given the level of where we are with language model right now, we could try to get the answer of which protocols are used in each publication, so that we can identify okay, is this a face related protocol for emotion or not, and it could give us at least a broader view of okay, which publications use which protocols. But I'm thinking in ways where you would do this automatically, and I guess that prompting a language model could be the best option right now, because you want to at least annotate in some way each publications to get a distribution of which protocols are used, and obviously observe the underlying bias. I don't have a better answer at least right now to this.
Yeah, I'm not sure if there are other ways of doing this, but thank you. Another question I have, if like in image-based meta-analysis, where we combine statistical maps that look at specific contrast, and like, for example, just using a GLM approach, then there are several scaling issues that may impact the unit, that may impact the results and poses a real challenge in image-based meta-analysis. So, for example, you have the scaling aspects that can impact the units of the contrast. So this will typically not impact the test results, but only the units of the contrast estimates. So if I understand correctly, in your work in training the model, you have combined Z maps, T maps, and beta maps. Does the scaling also potentially have an influence here or not?
So there is a preprocessing that is applied to each maps individually that normalize there are two steps. So there is a first, we remove the negative effects from the images, and second, we normalize the resulting maps, which aim to okay, what we look at in the end is on a given image, is there how large is the effect compared to the other distribution of activation, the distribution of activation in general. So I guess that obviously it must have an effect in some way, but we haven't measured specifically the effect. What we observe is that adding, so restricting ourselves to the Z maps led to poor results compared with using all the varieties of statistical maps that there is in Norway.
Okay. And so you, if I'm correct, you wrote that there was no influence, no real influence on the performance when you left beta maps out. Is this correct?
More than no influence is that adding T maps and beta maps in the training set helped to get a better decoding performance. And I think it's okay. What we are looking at is the distribution and I don't have a strong explanation for why exactly it's not an issue. Yeah.
Okay, thank you. Now, so in chapter 7, you propose peaks to image. You enable the creation of a database of dense spatial images. And so you wrote that publications often do not report statistical values associated with peaks. And I was wondering if statistical information was present. So I guess you mean the peak height. If that was provided, is this then taken into account or not?
Okay. So there are two. Sometimes it's also it's reported, but we are not able, speaking with PubGet, we are not able to extract values properly. So we made the choice to eliminate this information. Eliminate may not be the best term, but to not use this information, and rather to focus on the fact that okay, what we want is to use locations that are statistically significant in the context of the study, and given that it is the fact, we how do you say this? We count them into the process. Because the other issue is that if there is a statistical value that is reported, you also have to kind of know to put together the publications. You need to kind of know the number of participants and number of experiments. So it leads to it's feasible, but it needs a lot of processing of the publications, which we do not have, and we could spend time on this. But I guess that we had some other priorities, and I think that today I wouldn't spend time on this either if I had time. I think there's so much more to do on extracting the right information from the text of the publications, that handling the statistical values that is reported and that is subject a bit to the noise of how many participants there are and stuff like that. I think it's part of the meta-analysis process, and might be wrong. But I would focus, in terms of research, I would focus more on how to capture properly the experiments that are performed and what are each publication dealing with, rather than getting a bit more precise more precise idea of what the statistical value is.
Okay, thank you. So you mentioned this in your answer that when you would look at the peak height also, and then you would also have to take into account in some way maybe the sample size, etc. And I was also wondering about that. So if we know that if we only have peaks or reporting of peaks, that this may have several limitations, such as that different thresholding levels may have been used across studies, or smaller sample sizes, as you mentioned, can lead to the fact that only extreme peaks reach statistical significance and are reported about. And so there may be some kind of publication bias, and this can be aggravated by looking only at peaks. Do you think by only looking at peaks, so can you think that this will impact also the results in image reconstruction?
I guess that it affects in some way, but in our case, we master the peak extraction process, and we kind of are able to decide ourselves what are the statistical peaks that we want to extract. In particular, what we did during the training process is to add some variability to the threshold that we use to extract the peaks, so that so actually a given image in the training process is kind of duplicated with a variable set of peaks that are reported to it, to take into account the fact that, okay, if we outset the statistical value to 3, it might not be a proper representation of what scientists do. And we need to okay, have a variability here. So what we added different thresholds so that we kind of address this variability in the reporting of the peaks.
Mm-hmm, mm-hmm. Okay, so yeah, so in the training of your model, you have accounted for this, but then if you would go to the published peaks, do you think, so where you have no control over, do you think this has an influence on how, if you combine these data over studies?
Yes, so definitely. So I guess that for some studies it's a bit more impactful than from others. When we looked at the distribution of peaks reported in publications, and it ranges from less than 10 peaks to more than 200. And okay, so I guess that 1 in 200 is okay, it disappeared, but it's not an issue. 1 in 10 is a more an issue to get a clear view of the activation. So, but I mean, there is little we can do here. So, apart from publishing the full statistical image, which is encouraged, I guess that it's the main answer. So today there are no issues with sharing gigabytes of data. It wasn't the case 10 or 20 years ago. So now I think that okay, it may add a bit to the process, but makes sense for people to share those data because first it shows that there is some kind of reproducibility in what they do, and they're willing to open kind of what the experiments a bit more than just sharing peaks. Yeah, obviously it's more useful in the case of image-based meta-analysis to have this representation rather than the peaks, which are subjects to the typically those elements.
Okay, thank you very much. I have no further questions.
Thank you.
Thank you very much. Now I'll give the floor to Jean-Rémy King, who is a CNRS researcher and at École Normale Supérieure, but currently also detached to Meta AI.
Thank you very much, Raphael, for this presentation. I also really much enjoyed reading the document because I'm not prepared this in advance. I found it extremely clear, the introduction, especially with very nice figures, and the text is very pedagogical. So I think you should be praised for this, and we can really see the progression of the works throughout the three main contributions from chapter 6 to 8. And it's also nice to see that the contributions do improve on the previous SOTA. If I had one main criticism I would raise is that there is I was missing kind of a big discussion section about the systematic issues with the approach or the biases that you may encounter. And so I'm going to try to target the questions around this. But before this, I had some technical questions, mainly out of curiosity, to try to understand a bit better some of your results. So in particular for this this slide here, could you just clarify whether the decoding performance that you have here is the validation or the test set is using left out studies, or it's using left out concepts?
It's using left out studies.
Okay. And so do you also have this kind of performances for left out concepts?
But it's a supervised diffuser. So how would you do it for a left out concept?
So if you have like a word embedding, presumably you can generalize to new concepts. It's some sort of regressions.
So it's so what we do is that we have n classifiers which share in space, and so I'm not sure what you're saying exactly because okay, we train each classifier on each sense. Okay, if you have an additional one, why exactly? How do you initialize the weights to go from this maybe compressed representation that you learn from other concepts into this new concept?
So I guess if you know the ground truth, these are multiple words. But if there were single words, you could say, can I try to predict the word embedding for each concept given the brain map? And if I have a new brain map, which is not using a concept based on a concept which I've seen in the training sets, how far I am to the if I make the predictions of the word embedding, what are the closest words around, and how far am I to the true concept?
Okay, so that's actually an idea that which is more related to the neural context part. So which is a clip model. Yes, you know about. And so yeah, this idea of applying zero shot kind of classifiers. So we tried with the current model, and it's not working well. And so I guess my hypothesis about two of them. So one of them is we trained the neural context model in the coordinates presentation of the data. So when we transfer to applying a zero-shot classifier on images, okay, there is a shift going here, which is not trivial. And the second one, which I talked about in the presentation, is that we train on big publications, and we apply it. In the case of the zero shot, we would apply it to a single word, which is the same. So we looked into a shift depending on the. Can we identify embeddings that come from, for example, from the body versus from the abstract? Mm-hmm. So there's a clear separation. So we apply just the PCA on those. But you can clearly identify the two clusters that are on the main component. So I guess that those are the two potential reasons why the zero-shot classifying didn't work, which was a shame because I would have liked to work it from scratch. But yeah, I guess that so there are two things that we are doing right now. The first one is combining kind of fixed image and neural context, so adding knowledge of images into the contrasted part. And the second element is reducing the shift that we observe in the embeddings. And yeah, the long run I have in mind since last year is can we do some zero-shot classification either by just typing a working memory and seeing results, or by typing more descriptive descriptions of tasks or of cognitive concepts, which would be kind of the target. So kind of we want to describe experiments, describe stuff, and see what's the representation that we will get for it.
Okay, and so relatedly, I'm trying to make sure I read this graph here correctly. So it seems to me that the more the concept occurs in the text, and the worse is the decoding accuracy.
Yes.
And so, how do you interpret this?
So I think that's directly linked to the hierarchies that we apply.
Okay.
Because what we observe is that the terms that are lots in the training sets They are parent categories. They cover a large variety of kind of underlying conditions. So when you try to say, okay, all those conditions are all perception or are all it's not working well because of there's too much of a variety of activation, and I guess that's why we see this effect where highly specific, typically motor tasks, which are left-right stuff, perform very well, and on larger, broader categories, we underperform, and we are not able to, which kind of reinforce the fact that, okay, we cannot assign a single word to brain activity, and we need longer descriptions and what we are seeing.
Okay, thanks. For the following chapter, I wasn't sure about the dice coefficient. So if I read correctly the manuscript, the loss that you use is an MSE loss, and the dice is only for the evaluation.
Yeah.
Why do you use a dice as opposed to just I don't know, an MSE or a correlation values, which would preserve the amplitude of the response?
So that's kind of I think the dice, it gives a clear view of, okay, are we catching up the good activation pattern? Because if you have a statistical value, if it's 7 or 11, okay, so the effect is here, we're pretty sure of it. And the MSE on those will have the same effect between 0 and 4, on 7 and 11 on term of error. And it's an issue because once you are above a given value, and the value, the threshold value is not clear, you're not really looking to improve. And I tried to do a kind of MSC, which is clipped, so that if you have if you're above a certain threshold, the error doesn't apply anymore. But I think, yeah, what we wanted is to catch the activity, to catch the activation pattern that we have, is the proper repetition, rather than having statistical values that can lead to, in the optimization process to be, in fact, in the optimization process, we use the MSE in the end, but we normalize the edge that but yeah, in the evaluation, we didn't want to do this, and so that's why we use the dice, and we use the dice at different threshold, so that okay, you see the co-activation, but not at the single point.
But this is why I'm wondering. So because usually you want to adapt the evaluation, or you want to adapt the loss rather, according to your evaluation. So I'm wondering why, if the dice you think is better, why you don't optimize on a dice loss.
That's a good question. Okay. I don't have I guess I guess it will be trickier to implement, and it's feasible. Okay. I don't have a strong answer.
Okay. On the next chapter, the neural context contribution, I did not get. I think you say it in the text, but you also mentioned it in the discussion during the talk. What you do exactly for the projection head? What I understood is that you average all the embeddings of all of the output tokens. Is that right?
Yes. So first we chunk the duplication, match the context size of the model. Then
which is how many?
Which is what's the question? Can't say.
Okay.
I think it's
because it's a 7B. It's a 7B, so it's something like several thousands. So it's not the whole publication.
10K tokens.
Okay.
I will propose this.
4,000.
No, 4,000 is the embedding space you get in the end.
I
But
think it's pretty large because they use a sliding.
But it depends. I guess it depends on which model, because now it's like people use 70K tokens. It's largely the whole publication.
At some point, it won't be necessary. But at the same time, yes, the question remains of models that expect such a long input, and then you pass a working memory as input. So okay, there is something to deal with. So that's why we took IBC descriptions with the hope that okay, the longer descriptive that we get would help to make the shift a bit less and to get the actual representation.
Right.
But yeah, so doing the mean over the chunk is not ideal. It has proven to work in practice. I think it's the future, so the context size will increase, so it won't be necessary. But the question of how to handle small text versus such large amounts will remain. And so we tried into it of having in the training set elements of different input, typically taking the abstract and the full content. The first step that is kind of easy in the sense that we know that the abstract potentially covers topics are inside the publication, so we know that the mapping should exist. Question is now we select subpart of the text, how to select them so that the mapping will still exist.
Right.
If you take bibliography, for example, it's not.
It could help. For this, I'm wondering, did you make sure that the tables that could contain the P coordinate are not embedded in there, because then it becomes a kind of a trivial.
No, the tables are aside. So
Okay.
don't get the text on the side.
Okay.
Coordinates on the other. But you have the caption of the tables.
Okay.
Which I think is fair.
I was wondering whether for this publication, whether in terms of from moving away from technical points, more from a use case, whether the goal shouldn't be the contrary. Like if you click on a voxel in the brain, you would like to have, I don't know, the methods or the abstract that would correspond to the protocol that should be done by someone to indeed activate this particular voxel specifically, as opposed to the contrary. Is this something, because you are in a retrieval case anyway? So
yeah.
It's,
I think it. I mean, we reported in our case this way, but I mean the model is trained in a contrasted manner. So when we evaluate, I evaluate it on the other way around, and we get similar results. I think that's okay. It's for example, for neurosynth, and neuroquery, I've been using it mainly in the sense that I want to know what is brain activity for specific topics and stuff like that. What I do is I type text and look at the resulting image. I think both are useful for different use cases, but and it's not an issue because the contrastive learning makes it worse in both directions.
Yeah, for retrieval, yes, but for you, if you want to generate a text, then I guess you need an extra brick.
Yeah, but I've seen some papers. My main limitations in terms of knowledge right now is you can predict an embeddings, for example. How do you generate a sentence back from this embedding? Because it's feasible to predict embeddings from the latent space. And I think that there are some papers which the token to work. It's called this way. But okay, maybe I mean, for the neuroscientific point of view, it's too challenging right now. I think it's a topic in the general language, I would say, and fun topic to tackle, I would say. But we already are struggling with imaging data, and so generating your scientific text from the embedding space, maybe for later.
Fair enough. Okay, maybe moving on to a more conceptual discussion point. So you did mention in the talk and through the discussion already several types of biases. So you have biases that comes from protocols. For instance, HTTP is a very large data set. He has a few protocols which could bias the meta-analysis. When we look at the maps, like for instance, the FFA and PPA are kind of negatively correlated, which is probably not a brain effect. It's probably just because many people use face versus house as a protocol and tend to have sort of a negative maps accordingly. You also have publication biases, like the syntax brain maps is too good to be true. When you look at syntax correlate in large data sets, you have like bilateral activations, not necessarily focus on broker, whereas in this you tend to have. So you do mention them, but I didn't quite get what we can do about them in the future. Like how in this whole enterprise we could end up with a meta-analysis which is robust to this type of artifacts or biases.
I think that this artifact itself, bias itself, is coming from scientists who try to reproduce experiments that already exist, and so the meta-analysis is a clear target for this effect. So I think that the question is, should the predictive models tackle this bias actively, or should the neuroscientists that perform experiments try to replicate some previous experiment in some way, but by leveraging different tasks so that we have a variety of experiments that not induce those bias. So, I mean, the automation meta-analysis field is exposed to neuroscientists reproducing even bad results from previous studies and finding and actually reporting activity because they want a specific activity in those regions. And my opinion is that we find those effects in the models, and that's good. But is it our role to reduce them, or more on the experimental side? So, I would say more on the experimental side. Otherwise, it's kind of tricky because you're kind of trying to aggregate knowledge from the literature, and then you're saying the literature is wrong, so I'm going to remove this effect. But so it's kind of a loophole where you're deciding what you should report in the end in the meta-analysis process. So I'm not sure it's kind of the machine learning part where we have to do it.
Is there a world where, and this is really speculation, but where this kind of approach could inform the neuroscientists on what should be done precisely to decorrelate some variables which appear to be too heavily correlated in the previous studies, or areas which have apparently not been sufficiently investigated, or tasks which have not been sufficiently investigated? Like, could this help in this way?
I think that so what we discussed about identifying protocols that are used for a specific topic, it could be useful because right now at least I'm not sure other people do. They have an intuition, but there is no official report of emotion tasks are those, and they are used in the literature in those proportions. And maybe that's something one could build to have kind of an idea of what are experiments that lead to a specific concept. But yeah, in the end, it's hard, I guess. As a neuroscientist, the more you know about a protocol, the less you should use it. That's a kind of a conclusion we could have, because it means that it's widely used and might have an effect.
We have time for one last question,
Please.
So I was wondering whether you could discuss a bit the relationship between your work and a very different approach, which is based on maybe a few individuals with a lot of data being recorded with naturalistic stimuli, which presumably can target not the whole population, blob or center activations, but can find distributed codes or really subtle effects. Like if you were to go in one direction or the other in the next years, do you see these two types of approaches merging with one another, or do you see them as parallel tracks, one with a dead end?
I think it's a kind of two different tracks. Because okay, so among the processing steps that are applied to the specific image, one major step is averaging and transforming each specific image into the MNI reference, which induces a horrible, in an individual perspective, it induces an horrible averaging, which is not really, you can't do people specific right after this. So I guess that this work is more about knowing general knowledge about the distribution of concepts across the brain. And I mean, the way I see individual specific elements and models is that okay, we definitely want to have a lot of data on specific subjects because that's where we can kind of reduce first the variability and the dimensionality of the problem and do tasks on do individualized applications from your imaging. And then the way to extend to the population would be, I guess, a main point would be doing some transfer learning in some sense. In the end, it's a bit like language models. Like you have one big language model that learn a lot of stuff, and then you will specialize to each a new individual. So each time. So yeah, I don't see how they could merge in some way. I think that this work can provide information be analyzed at the individual level. But yeah, I think there are still two different kind of applications, maybe due to this aggregation, which is quite
great. Thank you very much. Yes, maybe there are links between large language models and the brain. I've heard of people from this lab who decided to move away from neuroscience precisely because of this. Thank you very much for your presentation again and for this great piece of work.
Thank you, Jeremy. Maybe before I give the floor to the supervisors, I'll ask a few questions myself. So, as I said, I'm Pierre Swagenbaum. I'm research director at CNRS at Listen on Paris-Saclay University. Well, I also appreciated the very clear presentation that you gave, and also in the document, the background and methods that underlie the present work. So that's not my domain, but that's these first 4 chapters were really very useful to me. And I've been impressed by the way one can scaffold some complex apparatus to work over the uncertainty, variation of the data sources, and still succeed in performing meta-analysis automatically. Then I have some questions first about the parts of the text of the papers that are really used. So I understand that in the first experiments, that's title and abstract, right? And then later you have the body. Is it the case?
So
I mean, to search in the first experience, I think that's only looking for publications that are selected. So
we use the full content of the publications each time, and we have try to specifically, and we show you that depending on the input that you take from each publications, you have more and more knowledge of what you more and more ability to map to true activation.
Right. Sorry, this is for neurocontext. I was also referring to the earlier experiments where you retrieve, and it's true that it's in different parts of the work. The publications are selected according to keywords to fetch.
So in the first, so in the EndNote content. What we have is only annotations which are on your vote and which are usually tags. So we don't even have the publication that is related to the images. And in this context, what we use is the full content of the document. But we apply a TF-IDF over the full corpus of neuroscientific publications.
But when you say the full content of the document, that's the tags or the publication?
No, that's the publications. Because we are switching from using images from your vault to using publications in the literature. So the modality that we are targeting is different. So in the first case, we have the image with very sparse annotations. And in the second, we have a rich content, which is the content of the publications, but with the stereotactic coordinates.
Okay. And so then in both cases, and maybe that needs a comment by Jean-Rémy, would it be relevant to try to select some specific passages in the publication that would be more relevant to the whole process?
Yes, so I guess everything in the publication is not related to the activation. We haven't tried, for example, dropping some, because we could select, eventually we could focus on the result section. So Object does this automatically to split depending on where it has been published. So we haven't tried to split the content at least yet. I guess that selecting the good part, which is related to the activation, is the key element. And it is a way to, at least I see it as a way to potentially have a clear representation in the latent space on the textual side, and eventually target shorter text, but which are highly relevant to the activation which is reported. So so that could be a solution to reduce indeed the shift.
A more minor question about in the results also with TF-IDF, I noticed that there were also multi-word terms that were in the process. So how did you select these multi-word terms initially?
So actually what we did is that we used neuroquery tools, which was built by a former PhD student and actual member of the SODA team. And it is based on a specific vocabulary list, which are neuroscientific terms which are a bit curated. So that's why there are some bywords and triwords, which are expressions that are kind of highly relevant in our context.
Okay, thank you. In the discussion, you explained that using the hierarchy, the hypernyms, the more general terms are somewhat less well predicted. And I was wondering whether it would be possible to well, not try to predict them directly, but still try to predict the more specific terms, the more specific concepts.
Yeah, so that's what we do actually.
And then infer the more general ones.
Here, so what we, for example, here there is an emotion term here, which is pretty well, so 0.75 of AUC. And on top of this, more higher on the list, the emotional face recognition, which probably is a child of
emotion,
of the emotion recognition that I've lost. Okay.
Yes.
So yeah, we didn't when we did the reporting, we merged kind of all the terms, matter how high they are in the hierarchy. That way we report as well the distribution of how many there are in the training set. It's an indicator that. Yeah.
But for emotion here, is that term predicted directly?
Yeah.
And what I was referring to was maybe not predicted directly, but use all of its children, predict them individually, and then infer the prediction of the higher term.
We haven't tried that. I think it's kind of what Bertrand has been doing in a paper in 2018 with Gael, where they what they do is they try to predict face or not face, and then dig up this decoupling based on the categories. We haven't tried to perform so. So there is kind of knowledge which are shared because we do not do N classifiers, which are totally different. We have 2 or 3 dense layers, and most are shared between the classifiers. So the representation that we get in the final dense layer is used simultaneously to predict specific terms and more broad terms. So there is some kind of knowledge which is shared that way between the terms.
Switching to neuro context. So I saw that you tried to retrain the language model on the medical corpus. Well, actually on medical publications, but it didn't succeed. Was that done before the contrastive learning?
Okay. And how do you know? Because I don't think I talk about it in the.
No, well, it's written. Sorry, it's written. It's written on page 80. Page 80. Okay. I just discovered it actually during the because I didn't notice it initially. And since the publication is by one of my former PhD students, I skipped on it.
So we actually try to fine-tune the LLMs. So the issue is no context. One issue that we have is that we have a massive overfitting. So it's hard to get the loss to properly converge. And that's why we made the choice of freezing the language models, because in this case, we didn't want to have those 7 billion parameters to update. So that's why in your context, the language model is fixed. But we also tried to, so we have this corpus that is available that reports stereotactic coordinates, but we have a much broader corpus of neuroscientific papers, which contain knowledge which you can train, fine-tune LLMs on just generating again embeddings through next token prediction, generating embeddings that are just relevant to more neuroscientific terms. So we tried to fine-tune models on this corpus, which contains around 400,000 publications. And what we observe, so is on the right here. So what we see here is that so we used kind of smaller models than the 7 billion parameters. And what we did is that we use the GPT-Neo models that we fine-tune either at 125 million parameters or 1.3 billion. And what we report here is the recall metric on either the fine-tuned version or on the phase one. And what we observe is that the gain is kind of minimal here compared to amplitude of things we can do on the computation side. And that's actually how we understood that the embedding space dimension is quite critical, because what we see is that in the end, the gap between the two set of curves is passing from an embedding space which is 700 elements to 2000. And that's why in the end we restricted ourselves to 7 billion models, and we didn't try to fine-tune it because it's time expensive, resource expensive, and the gain is at least for it's not critical for.
I understand.
But I think there are actually application which did at least on medical data fine-tune of Llama 3.
Okay, I think I'll stop here. Thank you very much again, and give the floor maybe to Damien, then to Raphael.
I'm not going to ask questions. I think I tortured you at least weekly for 3 years. The only thing I'm going to say is that it was a pleasure to see you evolve from a very engineer mindset to a more scientific and curious mindset. I remember at the beginning we had trouble like getting you out to like testing infinitely architectures. And also something that was hard was to see how you played a piece in the team as a person, in which way you can, in a very smooth and very quiet way, assemble people around you and provide them with good humor and energy. It's quite remarkable, and I think that will be missed in the whole parietal team. That's it for me. Mind team, sorry.
Thank you. First of all, I would like to say that you worked on a project which is very dear to my heart, because I believe it's somewhere machine learning people can bring something to neuroscience. I'm not always convinced we are useful, but I believe here we can be useful for the community. And this is why I really like the kind of project you did. And so this was hard, of course, because of the multidisciplinary aspect of this work, but you effectively did strong contributions, I believe. And you also benefited from previous work from Jérôme Decaisse, Romuald Menuet also. So we've been standing on the shoulders of giants, as we say, and you acknowledged it, so that's fine. I think that to speak more about you, you are really a doer, somebody who likes to do things, to put hands in the code and get something out of it. With your engineering background, you had to learn all about neuroimaging during the PhD thesis. And at the same time, all the machine learning AI background was moving so quickly. So we had to be updated all the time. So that was a challenge for you, because we were dealing somehow with vision concept and language concept. We also had to jump between the two. I think you really had the intuition to use the set transformer ID, for instance, and you were also very proactive in trying different NLP models recently for the neuro context. And so we can always rely on you to get the best possible performance for your setup. So you like to have a well-posed problem so that you can really get the solution for it. As Damien said, you are really also a team player, so somebody who likes helping others in the team. I think you've been, for instance, collaborating very usefully with Fateme, with a postdoc working on the project. You produce commons for the team, for the community. You like to produce new code or resources which will be used by others. And you are one of the few students who did not reinvent the wheel. Students like to, rebuild the world when they arrive because they don't understand the code of others, etc. And you spend time figuring out what others had done and improving rather than redoing. And this is great. And very few people have done that in the team, I would say. So I would like to thank you for everything you have brought to the team. And I also would like to thank the committee for the discussion and all the suggestions that you gave.
Thank you very much, Bertrand. Both Bertrand and Junior are research directors at MIND-INRIA. I need to turn to the audience to check whether there would be a PhD someone with a PhD who would like to ask a question. Additional question. Most often there's no one who wants to kind of break the ceremony because I need to ask for it. If there's not, we can move to the next part of the defense, which is a private part. So I guess the best is for us to stay here.
Yeah, and people leave, yeah.
And then we will need to devoir vous demander de la salle pendant ce temps.
Angela, Lars, and Beatrice, I'm going to create a breakout room and I will assign you to it. And they need to see. Yes, you need to see.
Oui, il y a des bouts de cachet. Private room.
Non, ça suffit.
Par rapport à
ça,
il y a deux qui sont marqués comme présents.
representations for heterogeneous brain image sets and text. This was in line also with your manuscript, which very clearly presented the involved research questions and methods. The jury considers that you provided significant contributions to neural based on those coordinate and text. Your work demonstrates clearly how different modalities can be effectively combined with you contributed to data, you improved development for the university community. And for all of that, the jury unanimously considers that she deserved to be granted the title of doctor in computer science at the University of Manchester. And we congratulate you on your assignment. Thank you.
So thank you to all the members of the jury, to Lars and Angela for reading the manuscript and for spending time writing the report. Thank you for all of you being here in remote, taking your time in the defense. Obviously, thank you to Bertrand and Degan.
Maybe something you have to read the oath if you remember, so that might help you.
You are giving your back to people.
Oh,
Let me help. Yes.
No, thank you for the opportunity to do this work. Thank you for being really caring about the PhD student. I was not very communicative in the number of messages I sent and the updates, and you were here to come get some news. Yeah, that's a nice thing we can expect from supervisors. And obviously, thank you to the whole team. So I simply moved from the early of COVID to a really nice community, and that's really nice. And merci à ma famille, à Sandra.
Emotion is a difficult topic to decode.
Ok, alors, sur la deuxième page, c'est le nom des compétences. En présence de mes pairs, parvenus à l'issue de mon doctorat en sciences du traitement du signal et des images, et ayant ainsi pratiqué dans ma quête du savoir l'exercice d'une recherche scientifique exigeante, en cultivant la rigueur intellectuelle, la réflexivité éthique et dans le respect des principes de l'intégrité scientifique, je m'engage, ce qui dépendra de moi, dans la suite de ma carrière professionnelle, quel qu'en soit le secteur ou le domaine d'activité, à maintenir une conduite intègre dans mon rapport au savoir, mes méthodes et mes résultats. So thank you, Angela, Lars, and Beatrice, and for those in person, there is a pool. Thank you again for taking the time.
Thank you for taking time.
Thank you. It was fantastic.
Looking forward to more discussions.